Welcome back to Dr. Liu's VCE Biology. Today's topic is regulation of biochemical pathways, as presented in the 2018 VCE Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. 4 students performed a series of experiments to investigate the effect of 4 different variables on the rate of an enzyme-catalyzed reaction. In each experiment, the students changed one of the following variables: substrate concentration, pH, temperature, and enzyme concentration. After recording their data, the students displayed their results in a series of graphs, as shown below. Each graph is a line of best fit for their data. The students did not label the horizontal axis on any of their graphs. The next day, the students could not agree on which variable should be labeled on the horizontal axis of each graph. The students suggested the following options for each variable. Graph 1 illustrates that the rate of an enzyme-catalyzed reaction increases with increasing variable 1, reaching a maximum value before decreasing sharply. This curve is characteristic of the effect of temperature on enzyme activity, where the enzyme denatures at high temperatures, causing a sharp decline in the reaction rate. Graph 2 demonstrates a similar trend, but the curve is symmetrical around the optimal value for variable 2. This symmetrical behavior is typical of the effect of pH on enzyme activity, as extreme pH values on either side of the optimum can cause denaturation and a decrease in reaction rate. A key distinction between graph 1 and graph 2 is the shape of the curve following the peak. Graphs 3 and 4 show that the reaction rate initially increases with the respective variables and then reaches a plateau. This saturation effect occurs when all available enzyme active sites are occupied. At this point, the enzyme becomes the limiting factor, and further increases in substrate concentration will not increase the reaction rate. This also means that if the substrate is limited, an increase in the concentration of the enzyme cannot increase the rate because the substrate is already saturated with the available enzyme. It is not possible to definitively determine which variable represents enzyme concentration and which represents substrate concentration, as both would produce a similar saturation curve. The results confirm that variable 1 is temperature and variable 2 is pH. While our analysis showed that variables 3 and 4 could be either enzyme or substrate concentration, Sheena's specific claim that variable 3 is enzyme concentration and variable 4 is substrate concentration is consistent with our findings. Methotrexate is a drug used in the treatment of some cancers and an autoimmune disease, psoriasis, which affects the skin and sometimes the joints. In both of these diseases, cells grow rapidly. Methotrexate is structurally very similar to folic acid. Methotrexate works by inhibiting an enzyme that catalyzes the change of an inactive form of folic acid into an active form. The active form of folic acid is needed for DNA production. With a structure resembling folic acid, methotrexate can act as a competitive inhibitor by attaching to the enzyme's active site. This attachment effectively blocks the binding of the inactive form, slowing or preventing the reaction. The reduced binding of the inactive form of folic acid leads to a decrease in the production of its active form. As a result, DNA synthesis is inhibited, which ultimately slows down cell growth and division. Antibiotics are generally considered unsuitable for the treatment of psoriasis, given its etiology, as an autoimmune disorder rather than a bacterial infection. However, they may be indicated in cases where a bacterial infection acts as a disease trigger or when a secondary bacterial infection of the skin has occurred. This section examines enzyme kinetics and the competitive inhibitors of an enzyme as a medicine. It is worth 8 marks and accounts for 6.7% of the total 120 marks. Multiple choice questions. 1 mark question 7. Enzyme kinetics analyze the effects of temperature, pH, enzyme concentration, and substrate concentration on the rate of enzyme-catalyzed reactions by matching these variables to their characteristic curves. Short answer questions. 7 marks question 8a and 8b. Competitive inhibition. Understand how methotrexate acts as a competitive inhibitor by mimicking folic acid. This action blocks an enzyme, inhibiting the production of the active form of folic acid, which ultimately stops DNA synthesis and cell growth. Question 8c. Role of antibiotics. Explain that while antibiotics do not treat autoimmune diseases, they can be effective in specific cases where the disease is triggered by or complicated by a bacterial infection. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.